fan of memorizing things just for the sake of memorizing them, especially when you can just like Google an equation. But there are some things in biochemistry that I think are worth memorizing. Not to be mean, but so that you can have conversations, you can better draw connections, and you can have this like flow of ideas without having to constantly be Googling something. So here are the things that I think are worth memorizing. Though of course, if you're taking a course, what you have to memorize is going to depend on your teacher as well. So these aren't things that you need to like go out and memorize from the very beginning. I mean, some of them won't really make sense until you've learned them. And so instead, these are things that you should be memorizing along the way and that I expect my students to memorize when we get to those topics in class. And so I'll break up this video and do parts of it in various times, but I thought I'd just lump it all together in this one guide for whoever out there wants to see it. Instead of just telling you what I think you should memorize, I want to tell you why. I think that giving it some context will help you realize that it's for your own benefit to memorize these things, and the types of things that you should be thinking about for the things that you're memorizing. So we'll go over some things like chemical names and structures, some things with core metabolism concepts and values to know, as well as some word parts. Knowing these word parts are going to be really, really helpful when you're trying to figure out what various words mean. And as I'll show you in a minute, I have a more extensive guide to word parts, but these are some of the core ones that I really think you should know. Okay, so let's get started. Amino acids are going to show up everywhere. Anytime we're talking about a protein, we're talking about enzymes that are proteins, we're talking about a chain of amino acids that's folded up. And so we need to be able to appreciate the structure of those individual amino acids and the fact that these different amino acids have very different properties that are going to influence how that protein folds and functions. And so we talk about amino acids a lot, and we often talk about them using abbreviations. So for each amino acid, it not only has a name, but it also has a 3-letter name. I like to think of it as kind of nickname, as well as a single letter abbreviation. So you kind of has like an initial and a nickname and then a full name. You should know all of these because there's like this 20 core amino acids, and we don't want to write out each amino acid each time. Say we have like an 800 amino acid long protein. We don't want to go and write each amino acid back to back to back to back to back. In addition to being really unwieldy, this is also going to be hard for computers to do things with because each of those amino acids is going to have a different length of the name, I mean. So what we do is we have these nicknames, and then we could do things like write a sequence where it would be MVA, which would be methionine, valine, alanine, rather than having to write methionine, valine, alanine. And so knowing these codes is going to then be able to, allow us to see, okay, well, if we see MVA, we know we're talking about methionine, valine, and alanine. And we should know that basically those are going to be nonpolar and uncharged amino acids. And why does this matter? Well, this matters because that's going to influence how that part of the protein might fold and function. Now, in order to be able to draw that connection between the name and the structure, we need to be able to put the structure to the name. And so I recommend, or like I'm going to require my students to be able to recognize the different amino acids. So if I show you this, could you tell me that's methionine? And then for my biochemistry class, I'm going to require that they also be able to draw that amino acid. Not to be mean, just because this is going to be really, really helpful at all times throughout the year, so that when we're talking about a protein, we're talking about a specific amino acid, you don't have to go and look it up. You can instead appreciate, oh, yes, that methionine is going to look like that. It's going to have that sulfur group, but unlike cysteine, it's not going to be reactive in that way. And so basically, you want to be able to put a name to those amino acids, know the properties of those amino acids, and ideally be able to draw those amino acids. So that was amino acids, but what about nucleic acids? Well, here we still need to know names, and it gets a little complicated because we have a couple different names for these different nucleic acids. And so when you're talking, I typically just talk in terms of like A, G, C, D, U, because that's easier, because then I don't have to remember which is the name of the nucleobase and which is the name of the nucleoside.
But part of the reason I have trouble with that, I think, is because I didn't memorize it well enough when I was first learning it. And so I want to make sure that my students know this so they don't have the same confusion that I had. So basically, when you go from a nucleobase to a nucleoside, you're adding a side of sugar and you're changing the name. So when you add sugar to adenine, now you get adenosine if you added a ribose, or deoxyadenosine if you added a deoxyribose. Guanine, you go to guanosine or deoxyguanosine. Cytosine, you go to cytidine or deoxycytidine. And thymine, you go to deoxythymidine. And uracil, you go to uridine. So for the purines, you're adding a sine, and for the pyrimidines, you're going to dine. So it's a little confusing, but you should know those names. Ideally, you should be able to recognize the structure and name the structures if you see them. So if you see like this, can you tell me that that is an adenosine? And so basically that would be like your ideal, would be able to see it and recognize it. Well, I mean, ideally you'd be able to draw it from scratch, but I think that recognizing it is a good ideal. At a minimum, you should be able to tell me if if something is a purine or something is a pyrimidine. And so if you have a purine, that's going to have 2 rings, and a pyrimidine is going to have a single ring. And you can remember which one is which in terms of which nucleobases or nucleosides are which, because the purines are Ag, the pyrimidines are Ctu, and you can remember the mnemonic pure as gold, which is basically saying the purines are adenine and guanosine. Guanine in terms of the bases, or adenosine and guanosine in terms of the nucleosides. See, it gets confusing when you talk about nucleosides, because then you also have to consider whether it's deoxy or oxy. So yeah. But anyway, you should at least know like the AGCTU and which one is which in terms of, is it a pyrimidine or is it a purine? And you should also know the base pairing, so the A to T or U and G to C, and you should be able to draw the corresponding base pairs. This is important because you want to know which of these nitrogens is going to actually be involved in bonding, which of those oxygens is going to be involved in bonding, which of them is free, which of them is involved in resonance. And so like being able to know those sorts of things and draw those hydrogen bonds is going to help you out. So those were our amino acids and our nucleic acids. And now let's talk about some of the constituents of those. So you want to be able to recognize both the structure and the shorthand, as well as be able to draw the following functional groups. So we're talking about functional groups. We're talking about these groups of atoms that have some sort of property. They're not just the carbon and hydrogen. There's some sort of extra bell and whistle that you put on typically you put on a hydrocarbon. And so you learn about these typically in organic chemistry. You learn a lot of things in organic chemistry. You learn some functional groups that aren't going to come in biochemistry, but these are the core ones that you're going to see in biochemistry, and you want to be able to recognize them, because as you go through, you're basically going to realize that these are going to go through similar reactions no matter where they show up. And so you'll find they have different properties, and then you can draw connections between them. And this is also going to allow you to kind of like have discussions about molecules and just casually mention, oh yeah, there's the ketone there, which is going to blah, 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 or there's the sulfhydryl group. And so I need to think about, is it going to be protonated or deprotonated? Can it act as a nucleophile? And all these different things without having to go and really look things up, but you can basically just know what the structure of that functional group is and its properties. And so the ones that are really important to know are carbonyl, so the carbon double bonded to an oxygen, a hydroxyl, so an OH group, also known as an alcohol group, an amine, an amide, carboxylic acid, carboxylate. So basically you want to be able to recognize, oh, these are just the same thing. One is just protonated and one is deprotonated. And often it's written in the shorthand where we have a COOH for carboxylic acid, and a CO minus for carboxylate. An aldehyde, so a carbon double bonded to an oxygen with a hydrogen on the end. We often abbreviate this as CHO. And if you have a carbon on either side, we have this as ketone. We also have an ester and an ether, as well as a thioester and a thioether, which are just the sulfur versions of this. A methyl group, so that's just a CH3, which isn't really a functional group, I guess, but I wanted to put this in here because it's often abbreviated as Me. An acyl group, that's basically where you have a double bond, a carbonyl attached to something. And in a specific case where we have this acetyl group, we see this a lot in biochemistry, and it's often abbreviated AC. Phenol, so basically when you have a benzene ring attached to something, we call it phenol. So you'll see this in like phenylalanine. And we often abbreviate it pH. And then a phosphoryl group, so a phosphorus surrounded by oxygens, we often abbreviate this with this like P in a circle. And you're going to see that all over the place when we're trying to add energy to molecules, make reactions more favorable. We often put a phosphoryl group on them, and so it'll be abbreviated. You'll have a molecule, and then it have a little stick hanging out, and it'll be on like a lollipop to a P, and that'll be representing a phosphoryl group. Okay, so by knowing these, remember, you're then going to be able to figure out, okay, what might a molecule do? How might this molecule react? By having an appreciation of knowing the makeup of that functional group. Okay, metabolism. So metabolism, see, there are those phosphoryl groups I was talking about. So we could have represented this if we're just talking about the molecule without drawing all of the atoms. We could have put a little like P with a circle, and that would be telling us, oh, this is like the energized version of a molecule typically. In terms of metabolism, there's going to be a lot of pathways, and I don't think that it's like crucial to actually memorize all these pathways. I think that what's important is being able to look at a pathway and being able to see what's going on at each step, being able to spot the differences and be able to tell, like, okay, which steps are generating energy, which steps require energy, why do we have this rearrangement, what type of reaction is this enzyme catalyzing? And so that's what I care more about than memorizing the actual pathways, being able to interpret the pathway. But I also think that it's important that you be able to recognize the components of the pathway. So I want my students to be able to recognize whether a given molecule is a constituent of glycolysis or the citric acid cycle. So the citric acid cycle is also called the tricarboxylic acid cycle. I typically just refer to it as the TCA for this. It's also known as the Krebs cycle, and it's kind of like a central hub of metabolism. And then glycolysis, this is where you're breaking down glucose. So there are a lot of metabolic pathways, but these are kind of like the two big ones that you typically cover in depth in a biochemistry course. And these are the ones that you hear about the most. Each of these has different molecular components and intermediates. And I want you to be able to recognize if I say a name of one of these intermediates. So if I said, say, glyceraldehyde 3-phosphate, I want you to be able to tell me that that's a component of glycolysis. And if I said oxaloacetate or malate, I want you to be able to know that that's a component of the tricarboxylic acid cycle. And so why? This is going to allow you to predict which pathways might be directly affected by changes in those metabolites, or like mutations in one of these enzymes in the pathway. What would you expect it to like directly influence? Recognize whether production of that metabolite requires aerobic respiration. So with glycolysis, we don't have to go through this isn't like aerobic, this is anaerobic. But the tricarboxylic acid cycle, well, here this is going to be going to generate these reducing equivalents, to go through chemiosmosis and the electron transport chain. So basically, this is going to be giving us our components for our aerobic respiration, and it's going to shut down if we don't have aerobic respiration happening. Whereas glycolysis, this is going to be anaerobic. And so if you have a component of the tricarboxylic acid cycle, well, then you're going to need oxygen in order for that to be going on. But glycolysis, this can be happening anaerobically. So by knowing if a molecule is in one of these pathways, you can then predict whether it could be happening under aerobic or anaerobic conditions. Also, it's going to help you see where building blocks for things like amino acids come from. So we talk about amino acid metabolism. We're going to see a lot of these molecules come up, and like alpha-ketoglutarate, we'll see that a lot. And being able to recognize that as a component of the TCA is going to be really, really helpful. This is also going to allow you to see where breakdown products of other pathways can join in. So glycolysis in its like pure form or whatever, it starts with glucose, but you can also have components from other sugars coming in later in the pathway. And similarly, molecules can enter the TCA at various points. And so by knowing which molecules can be in the TCA or in glycolysis, then you could recognize, okay, well, not only can I make this molecule somehow, but also I can put this molecule into one of these pathways if I make it from some other pathway, as long as the equipment's all, like the enzymes and everything is there. I also think that you should know like the starting and the end points of these pathways. So know that glycolysis starts with glucose and it ends with pyruvate, and that you get 2 pyruvates per glucose. Know that for the tricarboxylic acid cycle, you put in an acetyl-CoA, and that merges with an oxaloacetate. So oxaloacetate and acetyl-CoA are your inputs, but the oxaloacetate basically came from the previous cycle usually. And so those are your output is going to be oxaloacetate as well. And know like the net energy yields for these pathways. So for glycolysis, you basically have to spend 2 ATP, but then you make 4 ATP. So you end up with a net of 2 ATP, and you end up with 2 NADH, which can then go into the electron transport chain and produce energy that way. For the tricarboxylic acid cycle, here you're getting 3 NADH. Here you're getting 3 NADH, 1, 2, 3. You're getting 2, for the tricarboxylic acid cycle, you're getting 3 NADH, 1, 2, 3. You're getting 1 FADH2, and you're getting 1 GTP or ATP, depending on where the pathway is taking place. So those are your net energy yields, and it's helpful to know how much energy you're getting from these different things. Okay, some other values to know. In terms of like energy yields from oxphos. So you want to know like that FADH2 and the NADH, they come in different places in the electron transport chain. The consequence of this is that for each NADH, you're going to get about 2.5 ATP made because you're pumping out more protons, and then you have more protons to be driving this pump later on. And it's about 2.5 ATP that you can generate with FADH. With FADH2, you're basically coming in later in the pathway. You're coming in complex 2, so you're not pumping out as much protons, so you don't have as much ATP making power, so you only get about 1.5 ATP. So these numbers are helpful to know, as well as knowing why this is, so how they come in these different places. A couple other random values to know in terms of pKa's, so thinking about whether something's going to be protonated or not. Remember, the pKa is the pH at which half of it is protonated and half of it is deprotonated. And so a basic rule of thumb is that a carboxylic acid typically has a pKa of about 4 to 5. So if you're at a pH above that, you're very likely to be deprotonated. And if you're a pH below that, you're more likely to be protonated. And so typically we're at a pH above this. So the carboxylic acid is typically going to be protonated. In terms of a protonated amine, it's going to have a pKa of 9 to 10. And so if we're below that, then it's mostly going to be protonated. And so this is going to be helpful because it's going to allow you to do things like see that an amino acid is typically going to be in its Zwitterion form, where basically it has a protonated amine group and a deprotonated carboxylic acid group or carboxylate. One other value to know is an angstrom, this A with this weird little symbol. It looks kind of like a keychain or a loop for a necklace. This is an angstrom symbol. And 1 angstrom is equal to 0.1 nanometers, and we see this a lot in structural biology. It's a useful distance. Speaking of little distances, you should know like the smallifying metric prefixes. I don't know what the correct term is, but the ones that make them less than 1, they go below, or they go below to like smaller values. So you should know these down to like nano, ideally pico, and be able to convert between them. So you should know deci, centi, which don't come up that much, but you should know like milli and micro and nano and definitely milli, micro, nano. And so here's a little guide to how you can convert between these, and you should be able to convert between these without using a guide like this. But this is just to help you see how you can do it. Okay, let's talk about word parts. So as I mentioned, I have this much more extensive guide with lots and lots of terms, but some of them don't come up that much. So I think it's most important to know these ones that I have here. So it's important to know these because then when you see a term that you're not familiar with or a term is introduced, you're going to be able to know, oh, I think I know what that does without even having to do, or what that is without even having to look it up. And so you can basically predict what things might mean if you know those word parts. So some of these word parts are prefixes. So we talked about the metric prefixes, but there's some other ones that you should know. So in terms of numbers of things, mono, this is single or one, like a monomer, a single unit or a molecule acting by itself. Oligo, this is few or little. We can talk about oligonucleotides, oligopeptides. So when we're talking, when you hear the term oligonucleotide, oh, well, now you can think, okay, few nucleic acid chain, okay? Or oligopeptide, a short chain of amino acids. Conversely, you have poly, which is many. So if you have a polypeptide, well, that's going to be a long chain of amino acids. Hypo and hyper. So hypo is under or below. So I remember hypo, below. So if something is like hypoactive, it's going to have less activity than normal. Or something is hypotonic, it's going to have a lower tonicity. If something is hyper, that's over or above. So if something is hyperactive, it's more active than normal, or hypertonic, it has a higher tonicity. Then iso, this is equal or similar. So something's isotonic, has equal tonicity. If something's an isomer, it's going to be a molecule that's similar but differs in some way, such as having a different shape. And so you can see that we can use these prefixes in various ways to modify other word parts, and therefore, if we just see that word, we can then know what it means. Allo means other, so we'll talk about like allosteric, allosteric binding at another site, so not the normal binding site, or like an allosteric effect when something happens at one site in a protein and it like causes a shape change in another part of the protein and things like this. Endo and ento, this is going to be like inside. So remember endo, inside. Something is endogenous, say that's something that's made by the organism rather than taking in from the outside. If we talk about the opposite, we have exo or ecto. So remember exo, external. So something's like exogenous, it's like added artificially, such as like a drug you might take. Auto, this is self. So like an autotroph is an organism able to make its own food. If we're talking about like autophosphorylation, that's when a protein is like phosphorylating itself and that sort of thing. Inter, this is going to be between. So intermolecular interactions, this is going to be interactions between different molecules. So when we're talking about like intermolecular forces, we're talking about those molecules interacting with one another through things like hydrogen bonds, maybe some ionic attractions and this sort of thing. Intra, this is going to be within. So intramolecular interactions are interactions between different parts of the same molecule, like the hydrogen bonds in a protein backbone, as opposed to like the intermolecular hydrogen bonds that are taking place between the strands of DNA. Hetero, this is different or other. So if something's heterogeneous, it's made up of multiple things or not consistently distributed. If you have a heteromer, it's going to be a complex with different components. Conversely, we have homo. This is the same. So if something's homogeneous, it's going to be made up of a single thing. If it's a homomer, it's going to be a complex with multiple copies of a single component. Amphi, ampho, ambi, these are going to mean both. So if something's like amphoteric, it's able to act as both an acid and a base. If it's amphiphilic, it's going to have both hydrophilic and hydrophobic parts. Semi, this is going to be partly. So semi-permeable is going to allow some things through, but not others. Holo, this is whole. So basically that's easy to remember because it sounds like what it's saying. And a holoenzyme, say, is an enzyme with any necessary cofactors. Whereas conversely, we have apo, which is without or lacking. So an apoenzyme would be an enzyme without its cofactors. Neo, new. So gluconeogenesis is the making of new glucose from non-sugar parts. Some word roots. So gen or genesis, this is going to be like birth, creation, or making. So if you see like glycogenesis, well, glyco, that's one of the words that I took off of this list, but it's on my longer list. It's going to be like sugar. So glycogenesis is the making of glycogen, a storage form of sugar. You see hydro, this means it's involving water or hydrogen. So hydrolysis is the breakdown of a molecule by adding water. Oxy or oxy, oxy with a Y or an I, this is going to be involving oxygen. So if you see like oxidative stress, this is going to be stress that's due to the presence of high levels of reactive oxygen species. We talked about this a little when we introduced some of the functional groups, but if you see like sulfa or thio or mercapto, that's going to tell you it's involving sulfur. So for example, in the lab, you might come across dithiothriotol or beta-mercaptoethanol, which are commonly used reducing agents that contain sulfur. Last but not least, the suffixes. So you have philic or phile, which is going to be loving or lover. So hydrophilic would be water loving or water loved, and lipophilic would be lipid loving, lipid loved, and that sort of thing. Oppositely, you have phobic or phobe. So technically this means like fearing or fearer. But so like hydrophobic would be water fearing or water feared, but it's actually more like it's just water avoided than feared. And so more on that when we go into the hydrophobic effect. Lysis, this is breaking or breakdown. We saw that kind of when we talked about hydrolysis. If you see like catalysis, that's going to be the breaking down of biochemicals for energy or building blocks. Hemolysis, this is the breakdown of blood cells. So if you see lysis, you're breaking something down. And often that's done with the help of an enzyme. And when you see it, when the enzymes are often named in A's. So if you see this ASE ending, you're often talking about an enzyme. So for example, a nuclease is going to be an enzyme that cuts nucleic acids. So hopefully you can see how memorizing these prefixes and these word roots and these suffixes is going to help you be able to predict what a word means without having to just think about having to memorize tons and tons of words. If you know their core parts and what those parts mean, then you can get a good idea of what a word means, as well as predict sometimes what word would be the right word by knowing what that molecule should do. And so you can make flashcards, do whatever works for you in order to remember these prefixes. Some of them are probably pretty familiar to you just from like general use, whereas others might be new. But these are some important ones to know, and then see that more extensive guide if you need to know more. You can also do things like make flashcards for the amino acids, for the nucleic acids. I want to make like bingo cards, so we'll see how that goes. But anyway, those are some of the core things that I think you should know and you should memorize. And remember that the memorization you don't want to just memorize them for the sake of memorizing them, but because this is going to help enable a deeper understanding. So hope that helped.